All right, in this lecture, excuse me, we want to discuss some basic EEG features and parameters. By feature, I'm using this in the pattern recognition sense of the word. In other words, do we have some number that we can calculate from an EEG signal that would give us a way to look for what are often referred to as biomarkers, or ways that we can identify different groups as far as a disease process, or maybe different reactions as far as how the person is being stimulated. So in order to talk about parameters, it is important to talk about frequency bands. Most EEG parameters are determined from the frequency domain, but not all. Usually clinicians will look at a signal in the time domain and issue reports based on that. But researchers tend to look more in the frequency domain, especially when looking at data from a single channel. So we'll talk a little bit more about this in detail, but this is just to summarize what you've already read in some of the readings. This is to summarize what the different EEG frequency bands are. These numbers vary a little bit depending on which source you look at, especially in the beta and gamma. So you may see, you may see 24 here instead of 30. Some people will refer to a beta 1 and a beta 2. Main thing is, if you're reading or writing a paper, it is important to specify how you are defining it and to try to stay within the confines of how people in your field will report this. So for example, if you're working in Alzheimer's disease, important to look at the Alzheimer's disease papers and determine what, how are they reporting the beta activity. All right, this is a summary slide. We'll discuss each one of these in more detail. This is a summary slide for some typical parameters for the EEG. Not all of these are used as biomarkers, and some of them have several different ways of being calculated or presented. In this lecture, I'm going to present a basic overview because I thought it was important to have at least one video lecture outside of the readings where I discuss a few of the points that may not jump out at you in some of the readings or publications. We will not discuss all of these in detail, and this is not an exclusive list, so there are many others. We will have a presentation on EEG microstates, which is a completely different paradigm of looking at the EEG. So just be aware, this is just for this class, a summary of some of the parameters that we may or may not look at during the course of the semester. So the first, usually the first step, so you acquire an EEG. In this lecture, we're not going to talk a lot about filtering and things like that because that's for another lecture. But you acquire an EEG, you usually filter it with a low pass filter at about 0.5 hertz, and then you often filter it with a high pass filter, depending on whether or not and how high of a frequency you're looking at. The high pass filter can range anywhere from 50 to 100 hertz. If you're interested in looking in the gamma band above 50 hertz, then you also will need to include a notch filter for the 60 hertz noise. The more, obviously, the higher frequency you look at, there begins to be a question on the physical relevance of that, as well as the inclusion of additional noise. So the first step is to determine the EEG frequency spectrum and the EEG power in each frequency band, and the frequency spectrum is found through the Fourier transform. This is almost always done computationally, and it's usually a discrete Fourier transform because your data, by the time you get them on a computer, have been digitized. So determination, first of all, it's important to understand just a little bit of signal processing here. I know not all of you have an electrical engineering background, but you have to realize that there are some unique things about any signal that comes from the human body, particularly the EEG. So you have to understand that your frequency spectrum is always going to be an estimate. First of all, it's an estimate because the EEG is a non-stationary signal. In other words, it changes in time. You may have responses to things that are going on around you. You may have stimulation that comes in that your brain is going to respond to. You may have cognitive issues. Maybe you get bored, you start making out your grocery list. So the brain is not stationary, means stays the same with time. The brain is not really a stationary source, and so the EEG is, by very definition, it's a non-stationary signal. Now, we make assumptions, typically, often, that over a period of time, the brain state averages to kind of a stationary signal. But definitely, there are time localized events that if you simply take a frequency spectrum over, say, 10 minutes of data, will be averaged out. You also need to know that the EEG is sort of stochastic. So, what is stochastic? A stochastic signal is basically its properties are probabilistic and can typically also be modeled as the parameters can be modeled as having a normal distribution. Um, and so statistical models would represent a stochastic signal well. A deterministic signal means that you have some equations that could define and predict exactly what the signal will be. So, for example, the cosine of 2 pi t. That's a deterministic signal because there's an equation that tells you exactly what the signal will be for all times. Um, there are a few deterministic properties in the EEG. It's thought, because it is such a complex system with billions of active sources at any one given time within the brain. Many of them act in concert, and so approximations can be made. But it is believed that there may be some deterministic aspects to the EEG. The EEG is also most likely nonlinear, meaning that the differential equations that would define the state of the brain and the EEG at any one time are probably not linear models. They're probably not nonlinear models. However, as with stationarity, nonlinearity, we often make an approximation of linearity over a certain range. But remember, when we calculate a frequency spectrum, if you're an electrical engineer, you know that the signal has to be stationary, it has to be linear. And so as soon as we start talking about some nonlinear aspects of a signal and non-stationary aspects of a signal, we have to understand that the frequency spectrum is an estimate. Also, EEG recordings are time limited. So we have a start and a stop. We don't collect from minus infinity to infinity. If you're an electrical engineer, you know that the Fourier transform assumes existence of the signal from minus infinity to infinity. When you have a start and a stop, you have effectively inserted a window. You're looking at a segment of the data from time equals 0 when you turn the machine on until time equals however many seconds or minutes you're acquiring the data over. Now, the longer that time period, the closer we have to an infinite signal. But the truth is, they really are time limited. It's time limited by like a rectangular window that turns on and turns off in the time domain. When we take a Fourier transform, we're transforming not only our data, we're also transforming that rectangular window, and it has its own frequency characteristics. So however you choose to window your data, that is going to slightly skew the frequency spectrum data. So you need to be aware of that. MATLAB, along with some other signal processing toolboxes, basically automatically by default will take this rectangular window and they'll smooth the edges when they do the Fourier transform. So they use usually a different window shape unless you tell it not to. It'll default to something that has more rounded edges. Yes, this changes the data shape at the beginning and the end of the data record, but it is a less dramatic effect on the frequency spectrum, on the resulting frequency spectrum, than the rectangular window. All right, well, if we just take a standard FFT, these tend to have a lot of noise in the spectrum. So many individuals who do this kind of work use what's called a Welsh method. What the Welsh method does is it uses short windows in the time domain, estimates the spectrum, averages the spectrum of each window, and then it does overlap windows. So to clarify that, let's suppose you had 30 seconds of data. The Welsh method might take 5-second windows, find the spectrum for the first 5 seconds, and then shift the window instead of from 0 to 5 to be, say, from 2.5 to 7.5. Find that spectrum, then go from 5 to 10, find that spectrum, and then average all the spectra. This has the result of doing a couple of things. It minimizes any artifactual signals in the time domain, because what you can do is, if there is one segment of the data where the spectrum is way out of line with what it is everywhere else, you can assume there might be an error, do some kind of automated testing, and just throw out that particular window. But mainly the Welsh method has the effect of smoothing the frequency spectrum. Spectral modeling is another technique. This uses techniques to actually model the signal. Instead of finding the spectrum of the signal directly, you find the spectrum of the model, and then you determine this. So that also gives you a smoother and more predictable result. Okay, EEG power. There are several different ways to talk about EEG power. One is absolute power. That would mean you take a specific channel location and you calculate the RMS power within each frequency band. So you would basically filter your data for alpha, beta, gamma, delta, theta, and then within each filtered signal, you would calculate an RMS power that would give you the power in a given frequency band. Relative power can be defined in several ways. It can be defined with respect to the global power of the entire brain, which would be an average of the power over all frequencies over all channels, or it can be for a given probe. So I could take the EEG power at, say, location F1, and I could look at the power in the theta band, and then I could divide that by the power of the unfiltered signal, so the total power. And then, so band power over total power is one way to find relative power. This is sometimes used because in a resting state EEG, as you look across the brain, you find that there is a natural distribution of power levels. So if you want to compare, say, frontal lobe to temporal lobe in the beta range, it's difficult because during a cognitive task or a cognitive state, it can be difficult because the background EEG varies naturally. So this is kind of looking at what is the percent of beta power with regard to the entire signal. It's just another way of looking at it. Sometimes these terms are used in different ways by different people, so it's really important when you're reading people's papers to read very carefully and know how the author's defining their terminology. You have to realize that a lot of people who do this are psychology types, then you have people who come from an engineering background, then you have people who come from a medical background. Within each community, when people talk about things like relative power, they often mean different things. So very important to stay on top of that. All right, so this is just an example of what a digitized raw EEG signal looks like. And sort of what the steps would be. You would first filter for a certain frequency band. This is what a filter looks like. It's called a bandpass filter. It limits from frequency 1 to frequency 2 what you'll look at in this signal. And then once you've done that, you take the result, take the output of the filter, and you find its average power, RMS power. RMS, for those of you who don't know, means root mean square. And the actual calculation would be made in this way, where n is the number of points in your data record, and x sub I is the magnitude of each point. So you do that so that you don't have 0. Okay, the other method that we often use as a biomarker is EEG coherence. It's one of the many methods used to determine connectivity between brain regions. It's often referred to as functional connectivity because it's usually done it can be done in a resting state, it can be done during sleep. It's measuring from a sort of a physical perspective how well different parts of the brain are communicating with one another. So it's thought to be a measure of brain connectivity, um, as opposed to a structural measure, which would be if you put somebody in the MRI machine and you actually imaged the white matter tracts in their brain, and so you're actually physically looking at white matter tracts. That would be structural connectivity because you have actual structural data. Functional connectivity is more what EEG coherence is getting at, and that is, well, maybe you only have one white matter tract between point A and point B, but maybe it's working really well during a certain task, and so you're getting a lot of connectivity between the regions. Or maybe you have tons of connectivity, but if during whatever this person is doing, they don't need the connectivity between those brain regions, then the coherence might be low. So it is more related to the function of connectivity as opposed to the structure. The EEG coherence measures referred to as a normalized cross spectrum between two channels. Mathematically, the formula is given in the table that we looked at on the first slide. It is important to keep in mind that the coherence between channels does not directly correlate with the coherence between regional brain activity. What is this saying? This is saying that when you look at EEG power, you're getting an indication of the power in a given region. So let's take, for example, the frontal lobe and the parietal region. So if you look at EEG power in the frontal region and you look at EEG power in the parietal region, and then you look at EEG coherence, just because the power is high in both regions does not mean the coherence will be high, because you may have a lot of activity in that region, but if the brain isn't communicating within itself, you won't have a high coherence. So this is just a slide that shows that formula I just referred to. Here are two different signals, a signal X and a signal Y. Here's the formula for how the collection of formulas for how you would estimate both. Sometimes people refer to the cross correlation function. E is the expectation value of X and the expectation value of Y. Basically what you're doing is you're taking the signal and you're shifting it. You're finding what we refer to as cross correlation. You're then integrating that over all shifts and getting a function of frequency. The Rxy of tau is the cross correlation. The e to the minus j2 pi over tau is the Fourier transform part. You may, if you're from an electrical engineering background, remember that a power spectrum can basically be found by taking the autocorrelation and taking its Fourier transform. A cross spectrum can be found by taking the cross correlation and taking its Fourier transform. Do you have to memorize that for this class? No, but for those of you who have had signal processing or linear systems classes in the past, you probably know this. So the difference between a power spectrum and a cross spectrum is a cross spectrum is done between two different signals. A power spectrum is done between a signal and itself. And this little definition about the autocorrelation / cross correlation is sometimes kind of glossed over in some of the undergrad classes. All right, well, let's talk about what this means. These are some common coherence channel pairs. These are defined in one of the readings that I assigned. We can talk about bar coherence, which is often also referred to as long tracts. So in other words, this would be across the brain, and it's usually done from front to back. Anterior would be done in the frontal part of the brain between, say, maybe frontal and middle channels, and then posterior would be done in the back part of the brain with the occipital lobes. Here are the common channel pairs. This is done for the 1020 electrode configuration, and these are some of the ones that you will see reported in the literature. They sort of correspond to physical white matter, known physical white matter tracks between two brain regions. So I say sort of because it's kind of hard to say. People are only now really beginning to do very good white matter tract atlases. So unlike gray matter, which for example, we could tell you in any brain where the amygdala is, white matter has in the past been very difficult to image, and it's also very difficult upon autopsy to locate as well. So it's just been a little longer coming to get those tracks. With the advent of diffusion tensor imaging, we now have the ability to visualize in an MRI machine what those tracks look like. But we don't know which ones are used. So anyway, this is these roughly correspond to known connections. Okay, there's also some question of whether you look at posterior or back to front or front to back. So is coherence directional? The short answer is it can be. The generalized magnitude squared coherence, you basically form a matrix and then you find the eigenvalue of the matrix. This is believed to be more of a global measure of coherence, gives you one number so that you can talk about how coherent is this brain. May or may not be useful in a task-oriented environment. In a resting state environment, it might be more useful. And there are some publications that do refer to the generalized magnitude squared coherence. Here's a summary of the equations. So I just want to talk a little bit, I mentioned in passing this sort of global power where you would take power of all the regions of the brain and sort of average it across the brain, get one number for sort of like brain power. Same is true of the generalized magnitude squared coherence. These measures give you one number. One of the issues you have with EEG data when you have like 128 channels or even 32 channels, you generate many, many, many numbers. And for those of you who are familiar with clustering and pattern recognition algorithms, you know that you have all these numbers and you have to pick which one is the best at classifying group A and group B. The more numbers you have, the more you tend to do what is called overfit or overspecify the problem. Meaning, you can come up with a set of biomarkers that for your particular set of data appear to work extremely well. Then you try to generalize that over a number of different studies by different investigators, and those features don't always represent every set of data. So you've overfitted your set of data. This is just one way to get a different number that doesn't involve quite as many parameters. On the other hand, because you are sort of averaging over a number of numbers, you can average out the relevant ones. Final thing I want to talk about, I'm not going to talk about wavelets in this particular lecture, even though it is included on the slides. I want to talk about, but we'll have another lecture where we look at wavelets. I want to talk about the statistical moment measures or Yorth parameters. These were introduced by Yorth. It was a way to quantify temporal parameters in the signals. These are not widely used. This is one of the papers included in your reading. They're not widely used, but they offer a different perspective. So the statistical moments are the mean, the standard deviation, and then higher order moment intervals. If you've had a statistics class that's advanced, you've maybe seen these. If not, um, it's sort of like looking at the variation of the variation of the variation. So the higher orders would be sort of like the deviation of the deviation, if that makes any sense. And mathematically, that is defined in one of the readings, and you can read about that in more detail. I have used these. I kind of like them because they do provide a little bit of a different perspective. They don't rely on linearity of the signal like your power-based, Fourier transform-based, and even wavelet transforms. That's a linear transform. It's assuming a linear system. The York parameters don't rely on that. So because they are a temporal calculation, they do rely on sort of a normal distribution. So it is still more or less a stochastic type model. The wavelets we'll look at, well, okay, so here are the formulas for the different moments. The three York parameters that are most commonly seen are activity, which is similar to average power, mobility, which describes a mean frequency, and complexity, which is the deviation from the simplest shape. The simplest shape being kind of like a sinusoidal appearance. So this can be used either for spontaneous or event-related response, and it is a descriptive system that's based on time instead of frequency. Now, the wavelets are a little bit different. Wavelets are used because it allows us to see both time and frequency domain. If you want to use a time frequency method, the easiest method is the spectrogram. On a spectrogram, which is basically how the Fourier spectrum changes in time. So you take little windows, calculate the spectrum, plot it, and then you have various windows that are shown. The problem is you have to pick an optimum window length, and there isn't always an optimum window length. So for low frequencies, you'd want a long window. For small frequencies, you'd want a very short window. So what the wavelet does is it allows you, based on scale, you can vary the frequency resolution and the time resolution across different scales. So you can use a long time scale to get better information on your lower frequencies. You can use a short time scale to get better information on your higher frequencies. A wavelet is a transform, exactly like a Fourier transform, only instead of the exponential e to the j omega t as the transforming function, you have a wavelet as the transforming function, and it's in two variables: scale, which is analogous to frequency, and time. This is just an example of the difference. So here's a spectrogram of a chirp signal. A chirp signal looks like this. So it starts out low frequency and then it gets to higher and higher frequency. This is signal. This is like signal content versus time. And so if you just plot a simple spectrogram, you can see it gets kind of hard. We get pretty good resolution here, but then as time goes on and the frequency is getting faster, you see we just don't really see kind of the whole picture of what's going on. On the other hand, if you use the wavelet, you can see that you get a lot of power at this lower frequency, which corresponds to higher scale early on, and then as time goes, that shifts and more of your frequency occurs at a lower scale, which corresponds to a higher frequency. So this is not a class on wavelets or even on signal processing. So I'll just kind of leave it at this picture so that you can see that basically when you have non-stationary signals where there are pretty dramatic frequency changes over time, that's really well illustrated using a wavelet, not so well illustrated using a spectrogram. So EEG Lab and other EEG processing packages often include a wavelet option. This is what a wavelet function looks like. There are many different ones. This is just one, and I don't remember which one we chose on this particular slide. This is an example of what a wavelet transform looks like, where remember decreasing scale corresponds to increasing frequency. Basically what scale does, it depends on the function, but it changes the temporal length and some of the physical characteristics of what we refer to as the mother wavelet. So that's just, you know. Okay, and there are lots of different kinds. Clearly what I had on the last slide was the DB5. And you can see they yield different results. So this one is super easy to use, but you can see you get this weird ripple effects, and that's artifactual, that's not real. You get a few of them with this one, not so many with this one. So that's just some different illustrations. Wavelet coherence is similar to frequency coherence, but it's done between wavelet transforms instead of Fourier. Because of the nature of the wavelet transform, you'll get a lot more data. You'll have a coherence value at each scale. So once again, you can calculate an unlimited number of patterns or of parameters, trying to find the one that gives you the best description of what you're looking for can be like a needle in the haystack. Now, with computational power, it is now possible to do the needle in a haystack approach and say, you know what, I'm going to let only the data determine what is significant in this experiment, in which case you're doing what's referred to as data-driven research. People will often do hypothesis-driven research, which will say, okay, I believe that the right frontal lobe is going to be more active in group A than in group B for whatever reason. So there you would confine your analysis to just data that would prove or disprove your hypothesis. There are advantages to both. Your hypothesis might be wrong. There may be an effect that's happening that no one has anticipated, and so there wouldn't be anyone who'd come up with a hypothesis that maybe it's not the right frontal lobe, maybe it's the left parietal lobe, and if you're not looking, you won't find it. So that's an argument for data-driven response or data-driven approach. The main argument against the data-driven approach is that you will overfit your data on your specific experiment. So at this point, I'm going to end the slideshow and end the lecture, because mostly I just wanted to talk a little bit about some of the things that you'll be reading about in the readings. I do not have a specifically assigned wavelet reading for the undergrad students in this section, in this class. I will have one for the grad students. Undergrad students are more than welcome to read that if that is of interest to you. So I'll provide the information, but for those of you who are in 4332, that will not be required.